Okay, so let's go ahead and get started. Welcome to day 5 of our virtual neuromuscular conference. Today we will be having our rapid research rounds. Before I continue, what I'd like to mention is today's webinar is available in English and in French. On your screen, you will see an interpretation icon. It looks like a globe. Go ahead and select English or French, the language of your choice that you'd like to listen to today's presentation. I'll pass it over to my colleague, Véronique Penchot. She is one of our service specialists in Quebec who will also explain this in French. Go ahead, Véronique.
Merci, Amira. Alors, bonjour tout le monde. Bienvenue à la 5e journée du colloque sur les maladies neuromusculaires. Merci de vous être inscrit à la présentation d'aujourd'hui portant sur un aperçu rapide des avancées en recherche. Donc, je voulais vous informer que cette conférence est disponible dans les 2 langues, tant en français qu'en anglais. Donc, pour choisir votre préférence de langue, là, vous n'avez qu'à cliquer sur le bouton en bas de l'écran en globe terrestre, en forme de globe terrestre. Donc, le bouton interprétation et choisir la langue désirée. Sur ce, bonne conférence à tous.
Thank you very much, Veronique. So today's session will be a little bit different in terms of the types of sessions we've had so far with the Neuromuscular Conference. Today's session will be your opportunity to hear about research and presentation, research that is recently funded by our seed grant competition and also partnered funding through CIHR or eRare. You will hear short talks from the principal investigators on these research grant applications. To learn more about our research grant competition, I'll pass it over to our Director of Research, Dr. Boito. Go ahead, Daria.
Good afternoon, everyone. We have such a diverse group joining us today. We welcome MDC friends, families, donors, firefighters, researchers, healthcare providers, and industry partners. As Dr. Osman mentioned, today's session will focus on research.
I just got a call. I have to deal with something for our renovation today, just about a delivery because
I think we have someone, okay, there we go. In the past couple of years, we have seen tremendous gains in health management and the availability of life-changing treatments for individuals impacted by a subset of neuromuscular disorders, which not too long ago were considered untreatable. But the vast majority of children and adults impacted by neuromuscular disorders do not have effective treatments available, and we need research to discover these next medical breakthroughs. MDC's research program plays an important role in filling essential funding gaps and promoting patient-centered research. We invest in research initiatives that focus on diagnosis, developing new treatments, and improving health management for neuromuscular disorders. Additionally, we support initiatives aimed at infrastructure and network building to help translate and accelerate these discoveries into practice, as well as to support medical and scientific training, resource sharing, and clinical trial capacity. MDC provides research funding through a competitive peer review process. This ensures that grants with the highest potential impact are funded. However, each year, due to limited funding, we're only able to fund about 35% of the grants that are submitted, leaving many research questions unanswered. Throughout the conference this month, as well as throughout the webinar series that we have held since the spring, you have heard from past MDC research grant awardees, like Dr. Danielle Pierce and Dr. Amy McPherson from yesterday's Keeping Active session. And on Tuesday, you'll be hearing from another past awardee, Dr. Leanne Ward, about her research in bone health. And today, we will be introducing the newest grant awardees in Muscular Dystrophy Canada's research program. So now I would like to hand it over to Dr. Robin Parks, who is the chair of MDC's Medical and Scientific Community. He's also a senior scientist at Ottawa Hospital Research Institute. He will be speaking to us about research at all levels. And over to you, Dr. Parks.
Thank you. It's a pleasure to be here and to be able to talk to you today about the importance of basic translational clinical research. So just by way of disclosure, as mentioned, I'm the chair of the Medical and Scientific Advisory Committee for Muscular Dystrophy Canada, but I also am a member of the Scientific and Medical Advisory Committee of ALS Society of Canada. And what I'm going to start with is basically to just highlight what the take-home messages are going to be from my talk. So really what I hope you do to get out of it. First, there's different types of research tell us different things about diseases, and all of those types of research are important. Research is most impactful when we work together. And finally, research discoveries take time. So I'm a basic science researcher, and so what I do is I work with animal models and with tissue culture models of these neuromuscular diseases. And using these models, we can really develop exquisite insight into the disease at the level of the organism, the cell, and also the molecule. So we can really delve into what's going wrong within the cell and try to basically use that knowledge to come up with new therapies to potentially treat these neuromuscular disorders. We also can use these animal models to test these therapies, both for efficacy to see if they work, and also for safety before they ever move forward to human clinical trials. On the flip side of this is my clinical colleagues who really spend most of their time working with human patients. And really they provide the crucial insight into manifestations of the disease. So they really show us what's going on in the human condition. They can also do research to develop improved ways for screening for these diseases, new diagnostic techniques, and disease symptom management research, all of which are crucially important to improving the lives of those affected with neuromuscular disorders. And also importantly, they can test experimental therapeutics and devices to try and come up with new therapies to treat some of these devastating diseases. And really, the most impactful research probably comes when the basic researchers and the clinical researchers work together. And this is the gray area in between, which is called translational research. And the idea behind this is that perhaps we could take some of these insights that we find in these animal models and tissue culture models and some of these therapies we come up with in the lab and move that forward into clinical trials by working with our clinical collaborators. And this is called bench to bedside research. So things that we discover at the bench, moving it forward to the clinic, to the bedside to hopefully provide benefits for patients. The flip side of that, of course, is taking some of the clinical findings, so with these new diseases and possibly some of the tissues and the blood samples that we can get from these individuals, and having them analyzed in the basic science lab to try and improve our understanding of these various neuromuscular disorders. And this is called bedside to bench research, taking things from the clinic and trying to bring them into the basic science labs to improve our understanding of these disorders and again, potentially come up with therapies. And it's really an iterative process where we can combine with the basic science and the clinical research to try and move research forward. And really the idea behind this is that truly impactful research requires collaborative research teams. So that the days of a single researcher looking down a microscope and coming up with a truly impactful research are almost impactful research is almost over, because we're so specialized these days that we need to work with many different types of researchers, all that are experts in different areas to move the research forward. So whether that's a basic science researcher working with a clinical geneticist, but more often you require more than that. So bioinformatics specialists, so people that know how to manipulate and interpret large amounts of data, physiatrists, diagnostic imaging specialists, and many other people. And really it's the creation of these multidisciplinary research teams that will move the research forward. And Muscular Dystrophy Canada, of course, is very understanding of that and very involved in that. And they were involved in creation of the NMD4C, which is the Neuromuscular Disease Network for Canada. And you're going to learn more about that later. But I just wanted to highlight that MDC and this network are really designed to bring researchers from across Canada together to discuss and work together to try and come up with the new therapies of tomorrow. And the center is headed by Dr. Hans Lockmuller, and we're really lucky to have Dr. Lockmuller. He's an internationally respected rock star in neuromuscular disease research and clinical care, and he's heading up the network. And as I mentioned, you'll be learning more about that later. The last thing I want to touch upon is that research discoveries take time. And the example I'm going to use is the timeline for development for nusinersen or Spinraza. Now, nusinersen is the treatment that's available for spinal muscular atrophy. And you're going to learn a little bit about spinal muscular atrophy in the next talk. But I really just want to go over sort of the timeline for development of that particular therapy. So the first clinical description of SMA, spinal muscular atrophy, was in 1891. And it took 100 years before we were able to discover the causative gene. Now, much of that, of course, was the technology. We simply didn't have the technology in 1891 to be able to discover genes. But in 1995, we discovered the gene. And it took a few years to discover how that gene actually caused the disease. So what was the underlying molecular mechanism of disease causation? And that allowed us to develop animal models that faithfully recapitulate what the human condition is and start testing some of these new therapies that really address the molecular mechanism of this disease in the animal models. And so the first study for nusinersen in mouse models came in 2010. That was followed fairly quickly with human clinical trials. And finally, the treatment was approved in 2016. So that was basically 20 years from gene discovery to therapy. And I would argue that's actually relatively quick for a disease like spinal muscular atrophy, but that's about the time frame that we're working on when we're working in the labs and trying to come up with new therapies and understanding of these diseases. And Muscular Dystrophy Canada really supports over 150 disorders. And what I would argue is that for many of these diseases, we don't know the causative gene. We don't know the molecular mechanism of the disease. We don't have an adequate animal model to test therapeutics. We don't know the natural progression of the disease in patients, and we don't know how best to manage disease symptoms in these patients. And these are the types of questions that Muscular Dystrophy Canada is interested in supporting through their research grants, is to uncover some of these unknowns. And it's hoped that research funded through MDC will ultimately enhance the lives of those affected with neuromuscular disorders, uncovering effective treatments or cures. So we're going to hear about some of these, some of the great work that's being done in Canada to address some of these questions and uncover some of these unknowns. And the ultimate goal, of course, is to take today's research, whether it be basic translational clinical research, and turn it into the therapies of tomorrow. With that, thank you very much.
Thank you very much, Dr. Parks, not only for a great presentation, for your ongoing support with our Medical Scientific Advisory Committee. We'll pass it over to Dr. Rashmi Kathari. Dr. Kathari is the Deputy Scientific Director and Senior Scientist at the Ottawa Hospital Research Institute. Over to you, Dr. Kathari.
Thank you for that, and thanks for the invitation to present some of our work that led to the grant application to the Muscular Dystrophy Canada. I'm just going to change this so that the pointer will work. Okay. I'm going to talk about sort of a new sort of mild model for SMA, and I'll explain to you why that's an important thing to pursue. Here's my conflicts. Now, just to step back a little bit, what is spinal muscular atrophy, and why do we have spinal muscular atrophy? Well, really, from a genetic perspective, it's due to a mistake in splicing of the gene for SMA. And if you just kind of use a very simple sort of example of this, so here are 4 words or 4 components of a sentence: the let's eat, a comma, grandma. And in genetics, you need proper processing of this information, and proper processing will be let's eat, grandma, as opposed to when there is a mistake and important information is left out, it becomes let's eat, grandma. You can see how the difference in the end result is that the first one is normal, it's fine, it's all good. And that's when you have full-length SMN, the protein that is implicated in the disease. The second case is when you have the bad things happen. This is not a good thing, and this is because we have missed out on important information from the final protein that you'll often be referred to as delta 7 SMN, the truncated protein that comes from the SMN2 gene. And this is why we have SMA. It's really a disease of depletion of the full-length protein SMA. And so SMA is a motor neuron disease with muscle atrophy. Really what happens is that motor neurons undergo degeneration and their connection to the muscle is disrupted, and you end up with muscles that are not innervated that eventually atrophy, and that's spinal muscular atrophy. And the complications that arise from spinal muscular atrophy include scoliosis, episodes of pneumonia, there's difficulty with sleeping and nutrition. So there is really a need for respiratory, nutritional, orthopedic, and mobility support in this disorder. The other thing about spinal muscular atrophy is that it's really a disease that has a severity spectrum. And I don't have time to talk about why this is, but it's due to unique genetics. They're all caused by a single gene that's mutated. There is a more severe form, or the type 1, and then the intermediate form, type 2, and milder forms of type 3 and type 4, which are give you normal lifespan, and certainly type 4 is an adult onset form of the disease. A lot of what we understand about SMA at the pathogenesis level comes from our studying mouse models. As Dr. Parks mentioned, the first mouse model of SMA was developed 20 years ago, and that, I remember that day, and it makes me really old. But from that day on, there's been numerous mouse models of spinal muscular atrophy that have been studied to various extent, and really they served as a platform for drug discovery. And you all know about Spinraza or nusinersen, the gene therapies, Zolgensma, and most recently, the risdiplam or Evrysdi from Roche that shouldn't be on the shelves for treatment of SMA. Now, all of these were developed through studies on mouse models of SMA and really highlights the importance of basic science and the need for preclinical models. Now, the challenge that we decided to undertake is to model a milder form of SMA. And on this slide, I'm showing you that we were very efficient at modeling the severe end of the spectrum. We have many models of SMA at this end of the scale, where we have very low amounts of SMA and 5% to normal. We have severe intermediate forms where we can bring up the protein levels to about 15%. And then as soon as you get to about 30%, you essentially have a normal mouse or a normal with no real SMA phenotype. And to model mild SMA models, we have to really gradually increase the protein levels from 15% to about 25 or before you hit 30 to achieve this success. And the way we did that was to generate a mild model. And really what we did was take a severe model where we only had 15% of normal protein, and kind of like a rheostat, we're able to genetically induce a small increase in the SMN protein. And that's depicted here really. In a normal case, we would have this dark depiction of good amount of protein, which we can call it 100%. And the severe mice, we have about 10 to 15%, which is down here. And then we were able to gradually increase it up to about 25%, and that's in the middle. And so we've generated this mouse that has 25% protein. And dramatically, we were able to rescue normal lifespan in these individuals, mice, but they had shorter tails and they had reduced weight in this mouse model. And this is just some pictures at different ages, and they do grow old and a normal lifespan. But what we did observe was that these animals have motor function deficits. And we measured this by different ways in the laboratory. There is a treadmill for mice, if you want to call it that. It's like trying to stay on this rotating disc, and strong mice will be able to stay on, whereas the weaker mice will fall off, and that's what you measure. Or you can measure the strength and really look at both in the forelimbs and the hind limbs through a meter, determine what the grip strength is. And there are other tests like this that we can use. Using those tests, we were able to observe that in this new model of mild SMA, there were deficits in motor function, which was even more interesting was that these deficits were more prevalent in males versus females. Something new that is kind of emerging is the possibility of a gender or a sex bias in the disease, and really something that we would like to pursue further in the future. And on the basis of, what was the basis of this motor function deficit? Well, it's what you would expect. There is motor neuron loss, there are neuromuscular junction defects, and muscle atrophy in this mouse model. So I'm not going to go through the data, but really, it's really the hallmark features that you would expect from spinal muscular atrophy. So in summary, what we've done is we've met the challenge, we've modeled a mild form of SMA and have now filled in an important gap in our repertoire of animal models to study the full spectrum of SMA in humans. And so the future next steps for us is to fully characterize pathology in this mild model. What are the similarities to what has been observed in severe SMA? Are there differences that we might anticipate? Are there gender differences? Are these real? And can we translate those to human? And further, we really want to start to interrogate the molecular mechanisms underpinning the pathological changes in mild SMA. Again, the same kind of questions. Are the same pathways impacted as in severe SMA? And really like to acknowledge group members who participate in this work. And as Robin mentioned, this is really a team sport. Team Canada is central to our efforts. Muscular Dystrophy Canada is funding this aspect of our work, and as is Cure SMA Canada. But we also have international partners who fund us, but also collaborate with us on this project. I think that brings me to my time, and I'm happy to take a couple of questions.
Do we have any questions from the audience? Okay. Dr. Rashmi, as a established researcher in the NMD field, what would you say in the early parts of your career was the most, your involvement with the NMD community?
What was the most
meaningful? And
yeah, the critical part was actually my first grant ever came from Muscular Dystrophy Association of Canada, when it was known as MDAC. That gave me my start in Montreal when I started my laboratory, and which then led to grants from the CIHR. So the importance of the community and the charitable organizations in kind of triggering the research enterprise when I was trying to be independent was critical, and I'll never forget that. And since then, I've kept association with Muscular Dystrophy Canada in many different ways, being a member of their advisory committee and then a member of the board for a while. And I think that was really important. So for me, that was a highlight that I had to talk about.
Thank you. I see, I see a question coming in from one of our audience members. How do you translate the mouse results with only one gene SMN and 2 in humans?
Yeah, it's a good question from Joel. As you know, the genetics are quite different from human to mice. And so one thing we have a strong experience now is to model the human disease in the mice by actually using the human SMN2 gene as part of the transgenic components within the mice. And so the human SMN2 allows us to model the human aspects. And in fact, that's what led to development of some of the therapeutics that we now have, was the ability to put the human gene into the mouse context for study. In the short time I have, that's the best I'm going to answer for you, John.
Thank you. So hearing no other questions, we'll move on to our next speaker. If any further questions come in, we can also have some time at the very end, or answer them through our research hotline we can connect directly with the speakers. So next up, we have Dr. Luc Hubert. He is a professor of rehabilitation and a researcher at the University of Laval. His project is titled Muscle Strength Reference Values for Adults, a Critical Need in NMD. Go ahead, Dr. Hubert.
Thank you very much. Can you hear me well?
Yes. Okay.
Good afternoon, everyone. I'm very happy to be here today. Thank you for the invitation. And I would like to thank Muscular Dystrophy Canada for the grant received, which will allow us to advance our research work, which I would like to briefly share with you today. In everyday life, we do tasks that seem simple to us, and we are not really aware of what that requires at the physiological and biomechanical level. So to complete our daily activities, whether it is simply walking, climbing stairs, to get up from the sitting position, moving your wheelchair forward, transferring to bed, you need balance, you need coordination, and above all, we need a minimum of muscle strength. And having a minimum of muscle strength is even more important in the presence of diseases that affect our neuromuscular system. Once that has been recognized, the challenge is to establish force measurement protocols that lead to valid, reliable, and responsive measurements. So in other words, we need to develop high quality force measurements that will allow clinicians and researchers to better assess and treat patients. And for decades, there were two extreme ways of measuring muscle strength: manual muscle testing, and at the other end of the spectrum, the use of complex and expensive isokinetic device as the one you see here. But more recently, however, we have succeeded in miniaturizing the complex technology of isokinetic device in small portable dynamometers that allow manual but quantified muscle testing, as you can see here. And we now have a lot of experience with the use of these new generations of those manual dynamometers, to the point where we have developed measurement protocols which have excellent qualities and for which the influence of an evaluator properly trained would almost be zero, and the measurement error very, very small. And indeed, in previous studies with people with type 1 myotonic dystrophy, we have shown that using our standardized protocols, that muscle strength is an important biomarker, both from a clinical and research perspective, to document disease progression, for example, assess intervention effectiveness, or even guide prognosis. For example, we have shown that physical limitations in daily living activities, including mobility and especially walking, are related to muscle strength impairments, which ultimately lead to disrupted social participation. But what we lack in order to assess patients and treat them better are reference values of force to better assess the muscles that are affected and the severity of the damage. And that's what we manage to do in pediatrics. We have developed reference muscle strength measurements for children aged 4 to 18 years old that allow clinicians now to verify whether the measured muscle strength is within the expected values according to the age and gender of the child or adolescent. And we can even see on this muscle strength graph of hip muscles the gradual increase in values between boys and girls. And the comparison of these values, which become statistically different after puberty, so at the age of 13, 14 and above. So this is the type of data that we have not yet established in adults, data that would allow us to accurately identify the presence and extent of impairments in people with neuromuscular diseases. And this type of data will allow us to verify the effectiveness of certain short and long-term intervention. You may think about medication or therapeutic exercises, for example. But above all, with these data, we will be able to better target the interventions according to each patient. So this project, for which we have received a grant from MDC, aims to establish muscle strength reference values for healthy adults of 18 to 70 years old who are aging typically for 16 muscle groups from several areas of the body, including the shoulder, the elbow, the wrist, the hip, the knee, and the ankle. And we will also compare the variations of force between men and women and the profiles of muscle strength progression across age groups and between dominant and non-dominant limbs, as well as between upper and lower limbs. So we are looking at recruiting 300 healthy persons aged as I said before, between 18 and 70 years old, with an equal proportion between men and women. We will exclude athletes and competitive sports players. And we're looking at 16 muscle groups, again, at the shoulder, elbow, wrist, hip, knee, and ankle. And this project will be conducted in Quebec City and in Saguenay. And participants will have to attend one session of about 4 hours. So this is quite demanding for the participants. Now, to conclude, what will this project change for patients? To remain autonomous in our daily activities, we must have a minimum of muscular strength. And in the long term, the progressive loss of strength leads to physical limitations, which will in turn limit mobility and autonomy. Just to give you an example, in a previous research project, we studied the effect of a 12-week lower limb strength training program in 11 men with DM1. And we have shown that strength training induced an increase of muscle strength and lasting functional gains. So these results, of course, are very promising, but we are missing an important piece of the puzzle to progress. And these are the reference values of strength that we will obtain with this project. Of course, this project is part of a broader research program on neuromuscular diseases. And as you can see here on this slide, it brings together clinicians and researchers from several universities and research centers. All of these people are valuable colleagues to me who are involved in this project, and we will continue to work together to do all we can to improve the care of people who have neuromuscular diseases. So thank you very much.
Thank you very much, Dr. Bear. One question that perhaps you could respond to, it is a question that's in the Q&A, but I'll maybe expand on that question. You are looking at measures and you have this large age group in healthy individuals. I know you mentioned 18 to 70. Even within a same disorder for a neuromuscular condition, there's such a broad range. Can you speak to that? This individual is talking about themselves having Becker muscular dystrophy and their nephew having Becker muscular dystrophy. How can the same defect cause different symptomology? Can you speak to a little bit more about that, and how would that translate to muscle strength?
It's a very good question because we don't know much about all neuromuscular diseases in terms of mapping, in terms of impairments. For example, if we look at myotonic dystrophy type 1, for many years we kept saying that mainly these people were having distal impairments. It was simply because we were not able to properly measure very strong muscle groups like at the hip. Nowadays, we can do that. So what we have shown lately is that the severity and the range of impairments at the hip is almost as much as at the ankle. So I think we need to map more individuals with a diversity of diseases, and that's why we need reference values. We need reference values to be able to compare across diseases and across age groups. And when we'll have that, then we'll be able to do as we do in pediatrics. We could even have a better idea of a prognosis about we can anticipate over the next 5-10 years, what would be the loss of strength and what are the muscles that should be targeted to make sure we maintain mobility and autonomy of those patients.
Very good.
That answers the question, but
no, that was excellent. We look forward to seeing your results, and hopefully next time we can invite you back to share those findings with us. I'll go ahead and ask you to stop sharing screen so that Dr. Tétrault can share her screen. Dr. Martine Tétrault is an assistant professor at the University of Montreal. She'll be talking about her project, Identification of Modifiers Underlying the Variable Phenotypes of RYR1 Myopathy. We're really glad to have her here today. She has also been a past webinar panelist for us, and we
Thank you very much for the opportunity to present this project. So as a new investigator, I started my lab less than 3 years ago. So the opportunity to apply for funding in organism like Muscular Dystrophy Canada is very helpful. So I'll be talking about a project that we are putting in place in my lab, which is the identification of modifiers underlying the variable phenotypes of R1 myopathy. So this project is
I'm sorry to interrupt. How about if you change the display settings just to full screen? That way everyone on their different devices will be able to see your screen. Yeah. That will be excellent. Sorry. I apologize for interrupting.
No problem. Let's just see. I'll just stop share and start over again. Is it okay now?
Yeah. Excellent. Thank you. Go ahead.
So for this project, we are targeting patients that are affected by R1 related muscle diseases, but we hope that the results that we will obtain can be translated to a larger group of neuromuscular diseases in the future. So the main goal of our project is to understand the phenotypic variability that we observed in many neuromuscular diseases. As it was raised in one of the questions in the talk earlier, we can see different clinical presentation in patients that have the same disease and carry the same mutation. And with this project, I'm particularly interested in disorders that are related to mutation in the gene RYR1. So mutation in RYR1 is a gene that codes for the ryanodine receptor 1 and is associated with a wide variety of phenotypes ranging from malignant hyperthermia susceptibility to more severe diseases such as central core disease or neuromuscular disease, congenital neuromuscular diseases. So it is essential to increase our knowledge pathological mechanism that is involved with this variability, as it could help in patient management and also improve prognostic for those patients. So phenotype variability in RYR1 has been associated with the location of the different mutations. For example, mutation that is associated with malignant hyperthermia susceptibility tend to be located more in the N-terminal region of the gene, whereas mutation associated with a central core disease is more located in the C-terminal region of the gene. And since the gene RYR1 is really important for the calcium entry in muscle and subsequent muscle contractions, we can easily see that mutation in this gene have an important, the defect in the gene will impair the muscle function. However, the location of the mutation can partially explain the interfamilial variability and do not explain the intrafamilial variability, meaning patients in the same family that are carrying the same variant and presenting different symptoms. There's another study that was focusing on recessive RYR1 myopathies, and they saw changes in the transcriptome, so the expression of different genes, as well as the metylome, which are epigenetic changes. But when they look in dominant RYR1 disease, the results were not consistent, with some patients showing the same changes, but others having no change at all. So with these data, we brought the hypothesis that changes in the transcriptome and the epigenome could explain the phenotypic variability, especially in dominant RYR1 diseases. So when we want to look at the transcriptome, we're looking at the RNA from muscle tissue, and we are looking at two RNA species of interest, so the messenger RNAs and smaller RNAs such as microRNAs. So this is the first aim of our project. We also want to define epigenomic profile. So we will look at methylation, which are specific mark on the DNA that can have an impact on gene expression. And we also want to look at chromatin accessibility, meaning the way the DNA is open and accessible, which can be suggestive of active expression of genes. The third genome data together to really establish a signature, so a profile that could help us distinguish patients that have a malignant hyperthermia susceptibility versus patients that will develop a more severe muscle disease. So to make it simpler, so this is the molecular dogma that we have in biology, where we have DNA that will be transcripted in RNA, so just we're rewriting the DNA and then translated in protein, which the proteins will have all the functions of the human body. So in our project, our hypothesis is to actually that the fact that we have changes in the DNA code, so the DNA is like the blueprint if we wanted to build a house. So having an error or change in this blueprint will impact the construction of our house, which can be seen as a gene, the gene carrying the mutation. However, this particular mutation in this gene can also impact surrounding genes. So for example, an entire neighborhood impact the expression or the epigenome of these genes. There are also other DNA polymorphisms that are benign when alone they're considered benign, but when they're combined to this first change in DNA can impact surrounding genes. And we also have environmental factor that can impact the expression of the gene, of the other genes and epigenome. And altogether, this element can result in phenotypic variability and different clinical presentation, despite patients having mutation in the same gene or even carrying the same mutation. So far, for this project, we are recruiting a cohort of 21 patients from 7 families to answer the question that could transcriptome and or metallome changes explain phenotypic variability in ROR1 related diseases? So the patients we're recruiting have a wide range of phenotype from malignant hyperthermia to muscular dystrophy diagnosis. All the families that were recruited are from the Saguenay-Lac-Saint-Jean region in Quebec, which is a region that is known for this founder effect and that has a more homogeneous genetic pool. So the fact that we have a founder population and that they are all carrying the exact same variant make this cohort ideal to study the phenotypic variability. So the patients that are recruited are divided in 3 groups. So we have a group that is considered asymptomatic, which they have malignant hyperthermia susceptibility, a mild group where patients present myalgia and elevated creatine kinase levels, and a more severe group where a patient has muscle weakness and rhabdomyolysis. With those patients, we will get the muscle tissues, extract DNA and RNA, and we will use high throughput technologies for sequencing to get the profile of the microRNAs level, mRNAs, and also the metallome. And with that, we'll use the cutting-edge bioinformatic software and algorithm to analyze this data and produce signatures. So this research will have an impact. We think that the homogeneity of this cohort is really ideal to explore the mechanisms that are associated with variable clinical presentation. We want to look to see if those signatures enable us to stratify patient and inform on clinical management or prognosis. And we want to first focus on the Saguenay-Lac-Saint-Jean cohort due to this homogeneity to identify signatures that can be associated with different level of severity. And we then want to expand to a larger ROR1 cohort with patients carrying different variants and see if we can associate these signatures with a different clinical presentation. So this project is really integrated in one important part of my research program on neuromuscular diseases. In this program, I have two main projects. One is to identify genetic causes in unresolved NMD patients, and the second one in which this project is part of is to identify phenotypic variability signatures in patients. And for these 2 projects, we're using a similar approach where we want to look at the muscle tissue and look at sequencing approaches to answer those 2 questions. For this project, it's important to acknowledge the families that are participating, but also all the collaborators. So my lab, the clinicians at Saint-Justine, CHUM, and Saguenay-Lac-Saint-Jean that are participating, and of course, MDC for funding. So thank you very much. I'll be happy to answer any questions.
Thank you, Dr. Chitrul. The first question we have come in is if you could please explain what founder effect means and whether or not there are other disorders who also in Canada are more prevalent.
Yeah, so what we mean about founder effect is, for example, in the province of Quebec, so there had some what we call bottleneck that happened in when Nouvelle-France was founded. So first the French came to Nouvelle-France, so that this was the first bottleneck because there are really a few numbers of founders. We estimate around 6,000 to 9,000 founders that actually contributed to the genetic pool of French Canadian. So when we think we're 7 to 8 million French Canadian now, so there's a few founders. So this was the first bottleneck. And then when the English came to be established here, a lot of French Canadian moved further north in the province of Quebec, for example, in Saguenay-Lac-Saint-Jean. And this was the second bottleneck. So in those regions, the genetic pool is even less diversified, and it's not because they're consanguinity at a first level. So sometimes people think that this is due to consanguinity, but it's not. This is not the case. It's just that the number of founders many hundreds years ago were small, and there was not a lot of mix at that time. So French Canadian 100 years ago were not getting married a lot with English or other immigrants. So this has resulted in a more homogeneous genetic pool. There's a few diseases that are more prevalent in Canada, especially in Quebec. So there was one, sometimes it's the disease that are more prevalent, and sometimes it's a founder mutation. So it's a mutation that we found in higher frequency in Quebec or in certain regions. But there's also other example in other provinces in Canada where they have smaller community, the Amish community, for example, that could be where we see those founder mutations or those founder effect.
Perfect, thank you for that answer. So with that, we wish you all the best in your upcoming project, and we'll move on to our next speaker. Our next speaker is Dr. Reshma Amin. She is a respirologist and a researcher at SickKids. Her project is titled A Virtual Transition Innovation for Children and Adults Transitioning to Home Ventilation in Ontario, a pragmatic randomized controlled trial. Dr. Amin, if you can turn on your mic, please.
Hi there. Can you see the correct view of the screen?
We see the two screens, so if you can perhaps do full screen, that'd be ideal. Thank you. There we go.
Hi, good afternoon, everybody. Thank you so much for the invitation to present today. So I'm going to be shifting gears a little bit and talking about virtual care, which we've all come to become a lot more familiar with post-pandemic. And thank you for the invitation. I'd like to acknowledge Muscular Dystrophy Canada, who provided partner funds for our application for this randomized control trial. So just to disclose my grant funding. So individuals using home mechanical ventilation or ventilators at home, they use the healthcare system a lot, and they have a high associated burden of care. And it's important to note that neuromuscular disease today, both in children and in adults, is the most common reason for individuals to need ventilators at home. This population is complex in terms of their care because they cross many of the different healthcare sectors: home care, acute care, outpatient care, rehabilitation, and there's multiple different handovers in their care. Virtual care would be ideal for individuals who use ventilators at home. Just coming to clinic is very difficult because patients have to travel sometimes hundreds of kilometers to come to a tertiary care center. Just to bring all the equipment is difficult, and because of the medical fragility, even short transitions and travel become very difficult. There's multiple care providers, multiple different handovers, and largely there's a lot of barriers in terms of accessing respiratory professionals who specialize in ventilation. So virtual care is ideal because in children and adults with home mechanical ventilation, it would be able to bring the right people with the right expertise into the homes of these medical fragile individuals. So when we sought to think about a virtual care program for children and adults using home mechanical ventilation, we really want to try our best to get it right. And If that's the case, you have to go to the patients and families first. And we wanted a program, so we asked, we need something that's going to help to build confidence in patients and family caregivers, and how do we do that? And then from the healthcare provider side, with virtual care, you really want to make sure that you can get the clinicians the information that they need to support patients' needs. And this is sort of what we landed on.
Hi, I'm Cristiano. I like to go out and enjoy the world with my friends, but sometimes they can be really hard. Oh no, I forgot the post up. So my mom and doctor came up with an idea. What if we can doctor from home? A touch away. So now I can connect my doctor from home. She can ask me questions to see if I'm okay.
Cristiano, do you wake up at night with breathing difficulties?
Not at all. What's going on? And when there's a problem with my vent, help is just a phone call or touch away. Now I'm more time to play safely at home and less time going to the hospital. God! Hi, I'm Cristiano.
Okay, so our research question is, what is the effect of a virtual transition intervention delivered through a virtual care platform compared to usual care for individuals who are newly transitioning to home mechanical ventilation, with regards to the effect of healthcare utilization, so emergency room visits, hospital admissions, caregiver burden, cost effectiveness, and the efficiency of clinical encounters. So we had designed a randomized control trial. We are going to be recruiting new home ventilator, mechanical ventilation users from 7 hospitals across Ontario. We are going to be randomizing them in a 1-to-1 fashion. So there'll be a total of 440 patients to either receive the eHealth intervention or usual care. Our primary outcome was looking at emergency department visits at 6 and 12 months. And then in terms of secondary outcomes, we are going to be looking at healthcare utilization, participant and caregiver outcomes. We've planned an economic analysis, as well as looking at on the impact of healthcare providers using virtual care. And then as we follow these patients along, we'd also plan to do qualitative interviews with patients and families at 6 months and 12 months. But then COVID-19 hit. And so we had co-developed the Touch Away app for the home mechanical ventilation population, and we were all set to launch our trial in summer of 2020. But now with COVID, it became evident that it was now important now more than ever to keep our patients safe at home. So we pivoted and we partnered with the Ontario Ventilator Equipment Pool, and they funded a COVID-limited provincial rollout for all existing home mechanical ventilation users during COVID. So we've been able to provide the virtual care platform to children and adults who are existing, so already using ventilation in Ontario. The app is being used to support intensive care management, and patients have been downloading the app on their own devices, and a subset are given tablets if they don't have internet or if they don't have a compatible device. And we've been successfully able to partner with the 7 hospitals across the province. So how did we get here? We started off with a grant, then we hit the pandemic back in March. In May, we made the decision to pivot from our randomized control trial to a clinical rollout. We received funding through the Ontario Ventilator Equipment Pool to fund the clinical rollout. And as of November 2020, we now have 180 patients enrolled in the clinical rollout from 7 hospitals across the province. So what does this virtual care program entail? So it's not just a clinic visit that you can do virtually. So that is a component of it. So you can do routine virtual clinic visits. We also have the ability to do two-way communication, meaning that healthcare providers can initiate contact, but also families and patients can initiate contact with us. And it can be via text, video or telephone. We also have the ability to do remote ventilator monitoring, so we can monitor their ventilators remotely from their home. We have an educational resource library, so patients can access this at a touch of the button in terms of a refresher on how to use their ventilator, how to clean their ventilator. We've worked with patients and families to develop a virtual care plan, so all of their information and all their details are at their fingertips, and they also can update that information. And one of the last things that we've done is we've incorporated questionnaires that go out to patients and families so we can follow them. And depending on how they answer the questions, they can be flagged as being green, yellow, or red. And if they're yellow or red, it prompts a reach out by the clinical team. So this is what the interface looks like. So you can see at the touch of the button, you can either text or call or video call. The patients can do this, or we can do this as the healthcare provider. There's a virtual care plan, and so each of these different icons will open up into a form that then has healthcare information about their different respiratory technology types and all of their ventilator settings or cough assist settings, but also the medications and vaccinations. We also ask questions. We send out questionnaires. This is one example, of one of the questions. So in the past week, have any of the ventilator alarms been ringing off? And if the family answers yes, then there's a place for them to answer which one, and then we can reach out accordingly. We have live ventilator downloads that transmit information from Bluetooth. So we can see their ventilator statistics and also how they're using it. So we can follow our patients in real time based on their adherence with therapy and how effective the therapy is for them. And then we have this educational resource library where families can access information that's timely and up to date to address any issues that they have. So we're currently, we're continuing to enroll patients from the seven hospitals. We have 180 patients enrolled to date, both pediatric and adult, both English and French speaking. And I think what's really exciting is that we've been able to enroll some adult ALS patients who are nonverbal, as the app provides a way for them to be able to communicate with their healthcare team. Early sort of feedback from patients and families has been very promising. They talk about a sense of connection and feeling safe and secure, because it's even easier than before to reach out to their care team. And from a healthcare provider perspective, we're also hearing from our colleagues that there's been benefits, that it's very efficient and timely to be able to access the ventilator data in real time. It helps to facilitate troubleshooting because you can use the video conference feature and the three-way calls. So that's also been really exciting. So in terms of next steps, so here we are now in November 2020. We're aiming, if COVID cooperates, to be able to launch the randomized control trial. So the difference between the clinical program right now is that we've rolled that out clinically to all existing ventilator users, and the randomized control trial was targeted to those individuals who are newly starting ventilation. So we're aiming to launch that in February, and then we've planned an evaluation for our clinical program at the end of May of 2021. So I'd like to acknowledge my incredible group of collaborators, both for the randomized control trial and also for the clinical program. Also want to acknowledge Muscular Dystrophy Canada, because they provided partnership funding for our CIHR application, as well as all of our other funding partners. Thank you.
Thanks so much, Dr. Oman. Just being mindful of time, I'll probably limit this to one question, which is, it's interesting. The comments that are coming into me about this presentation are similar to the feedback that you've received and that you just shared, that qualitative feedback. And the most common question is this eagerness to see this in other provinces. You mentioned Ontario as the province where the clinical rollout happened. Can you speak to plans of expansion in other provinces in Canada?
Yeah, absolutely. It's a great question. I think that one that I'd be happy to hear about and sort of partner, because It's one thing to develop it in one province, but really, I think, we have to make sure that this can be rolled out across the country to provide access. And, whether you're a patient living in Ontario or British Columbia, your care is the same. And so this is definitely something that we're excited about and looking to work collaboratively across the country with others.
Fantastic. Thank you so much. All right, so I'll turn it over next to Dr. Benjamin Gallet. He is a clinical psychologist at the University of Sherbrooke. We'll be speaking about today empowering caregivers to better manage DM patients' neurobehavioral symptoms, myotonic dystrophy. Over to you, Dr. Gallet.
Can you hear me? Yes, sorry, I started too early. Again, thanks everybody from MDC and to give me the opportunity to present our research. And sorry to look a little bit at noise if I will start to talk about myself, which I usually don't do, but just to tell you a little bit of the background of this project. So. Yes. So I will talk about my professional journey only for facts on MAD, not to talk about myself. So I used to be a clinical psychologist in Paris in a great clinic for neuromuscular disorders. So this is why, I mean, it's important to me because all the research, all the projects I do now are always focused on how can I help, people in daily life. So that was my first point. And so anyway, I made a dissertation on myotonic dystrophy, the psychological adaptation. But then I moved to Saguenay, Quebec, Canada, which we heard a lot so far today. So so I joined the GREEN, which is a research group that is, it's very interesting. It is really connected to the neuromuscular disorder clinic. It's on the same floor. So, it's very exciting for research and clinical research. And yeah, so the GREEN. And why should I leave Paris for this very small area and not for the beautiful landscape? I mean, they are, but also because as we heard today, for DM1, I would say, unfortunately, the DM1 prevalence in Saguenay is very important, one of the most important in the world, So it's unfortunate, but it provides opportunity, of course, for research. So anyway, I stayed there, and it's been now, I don't remember, maybe 6 or 7 years that I'm staying there. So briefly, the research program in DM1 that I'm doing in relation with this group, led by Cynthia Gagnon. We have many projects in DM1, the characterization of the evolution of neurobehavioral symptoms. We also are working on validation of outcome measures, more specifically myself on fatigue, apathy, and cognition. Also, there is an ongoing project on the neurobehavioral and sociocognitive symptoms and their impacts on daily life, autonomy in the infantile phenotype. And of course, as Dr. Luque presented earlier, I'm part also of this project of the effect of muscle strengthening program, because we have observed interesting outcomes on neurobehavioral symptoms, actually. So many other projects in other neuromuscular disorders, but I will focus on DM1 today. So finally, the MDC funded project who started, let's say, July 2020. I'm very excited about this project, I actually have to say. So empowering caregivers to better manage DM1 patients' neurobehavioral symptoms. So what is this neurobehavioral symptoms that I will call the NBS so far, I mean, from now, because I couldn't breathe anymore. So anyway, in DM1, maybe you know that, but among muscular and physical impairments, there are many cognitive and behavioral manifestations, and they are related to the brain impairments, and this is why we call them neurobehavioral symptoms. So specifically in DM1, the cognitive manifestations will lead to lack of adaptive behaviors, lack of socialization, difficulties in communication, difficulties in daily living tasks, also problems with judgment and decision making that can lead patients in many trouble, in social situations that can be very difficult for them and their family. And there are also many behavioral manifestations such as avoidant personality, excessive fatigue, excessive daytime sleepiness, and apathy that we could describe as a reduced initiative, interest, and emotions. So NBS, they impede on patients' daily life, so sometimes if you ask them actually how they feel about the disease, they're gonna talk more about fatigue and daytime sleepiness than their physical impairments. But it also have an impact on family and caregivers too. Here are some quotes. So, for example, in the Anne Woodbury book, who's a lady living with 5 DM1 affected people in her family, she said that my children are always tired and lazy. Another example, it's what can be postponed was postponed. Sorry, it completely irritated me. So here we you can see how people can be impacted by these symptoms, and it could lead to family and social misunderstandings and conflicts that are very often reported. And of course, these symptoms are less known or recognized than physical manifestations. Maybe because they are sometimes you can't even see them, like cognitive or fatigue. You can't see it in the, actually, sorry for the expression, but on the face of the patient, So it is very difficult to understand, and it is very difficult to deal with it for caregivers and partners. And an example, again, in the book of Miss Anne Woodbury, she wrote, "Now that I know why my family are the way they are, I actually feel empowered." And this sentence is very important because, of course, this lady made all the efforts and had the education to look everywhere and to search for actually for information, but it is not everybody that can have this opportunity or this insight initiative. So what we are looking at in this project, the main objective is to provide to caregivers with practical tips. This is very important word, practical tips, aiming to better understand and deal with their affected ones, NBS, in daily life. So more specifically, the first objective is to identify concrete challenging situations where NBS interferes with daily life. And, we're gonna interview partners, caregivers of DM1 affected patients. Then these participants will identify with us effective and non-effective strategies that they use to deal with or to support their affected ones. And let's say that based on this, this identification, we aim to develop, again, practical advices, information, tips, products, in an adapted language. And this product could take the form of motion design briefs, cartoons, videos, web pages, anything that can be useful and, again, practical for caregivers. And this is a co-construction project because patient partners and the caregiver partners are involved throughout the project. All along the project within the research team. Each step will be made in collaboration with these researchers, let's say, you know. So again, it's a very applied project, and this is why I'm very excited, because we, you know, personally, I did many research to better understand these symptoms on a scientific level. But this is a great opportunity for me, and again, thanks to DMC, to you know, to transfer all this information, scientific information, but to transfer it in some very useful products or deliverables for people. I mean, you know, for caregivers, and of course, with an impact, I hope, on patients themselves. So thank you very much. This is it. I would be happy to answer the questions, of course.
Thank you very much, Dr. Gallet. A few of the comments that came in is they felt like you acknowledged some of the symptoms that they feel or have experienced. And also some family members also in attendance are thanking you for acknowledging them as someone who is also impacted by myotonic. And my question to you is, when we think about patient-oriented research, we're thinking about the individual, but your project highlights that you're looking at the caregivers as well. Can you speak to why family members should be involved in research like this?
Oh, you know, I mean, I think there is a general answer, but I will go first with the specific answers. I'm sorry, I don't see you, but I will try to to answer anyway. So yes, the specific reason why in this project is that, as you may say, in DM1, I mean, actually the cognitive symptoms sometimes may lead to, you know, to a diminished level of awareness of the disease. So, and more specifically for these cognitive symptoms, because of course, I mean, fatigue, daytime sleepiness, of course, patients will openly tell how much they suffer about it. But when you come to apathy, you know, lack of motivation, actually, this is, how can I say that, intricated, that they don't, sometimes the patients don't really realize that they lack of initiative. So the impact is even greater on caregivers, you know. So I think that we start with actually who is helping the patients in daily life to provide them, you know, support for symptoms that sometimes, you know, patients or affected people don't really actually have awareness of, you know. So this is one of the specific reasons why. And of course, because as I said, there is always opportunity to work with patients, but again, caregivers, they, as I said, they are impacted by the disease in a way, you know. And you know, this neurobehavioral symptoms, it's very, very important that because, you know, it's so invisible that we have to provide more information. But again, we will try to start with the perception of the experts, which are the people living with affected DM1s.
Excellent. Thank you again for this important work, and we look forward to seeing the product that you spoke about, and we'd be happy to share with the MDC community. I'll pass it over to Daria, who will be introducing our next speaker.
All right, our next speaker is Dr. Benoit Gentil. He is an assistant professor at McGill University, and his project is titled Therapeutic Potential of CK1 Agonists in Inherited Peripheral Neuropathies.
Thanks a lot for this introduction. I'm I'm going to be the science guy probably in this session, but don't worry, I'm gentle, so I will guide you through all of this, and it should be a smooth journey. So thanks a lot for the invitation. I'm very glad to join you. And here I would like to speak a little bit about my my work and what we are doing. Okay, so here we have. I would like to speak about hereditary sensory motor neuropathy, or the disease is also called Charcot-Marie-Tooth disorder. It's a disease that is very different from what we have spoken so far. Just to let you know that this disease is the most common inherited neuromuscular disease, and the prevalence is quite high. It's 1 in 2,500. What is this disease exactly? So it's a disease that was described a long time ago by Charcot and his student Mary and another physician Tooth. And it affects these neurons that connect your spinal cord to your muscle. Okay, so that's where everything happens here. So you have motor neurons going down to the muscle, or you have sensory neurons moving up to the spinal cord and bringing back the sensory information. In Charcot-Marie-Tooth disease, these two type of neurons are affected, and they are affecting the gene in general. The consequence is that you have an amyotrophy, so your muscle is actually being amyotrophic, and the characteristic is here, as you see, what we call the pes cavus, because the muscle become weak and the foot start to show some deformities. So it's a characteristic disorder. When does it start? It starts quite early, usually in your first decade of life. And now the trick with this disorder is that so far there's more than 100 genes that have been described to be causing Charcot-Marie-Tooth disorder. So it's a big challenge here to find out what is exactly happening with this disorder. And we detect two forms, one that is called demyelinating and another one that is called neuronal or axonal. So what's happening with these two forms? If here you have a characteristic neuron, okay, motor neuron, with the cell body and the dendrite. So the dendrite receive all the information from your brain. They integrate this information, and then your neuron send back a signal, send down a signal to your muscle, basically contract or not contract. And along this projection that we call an axon, that is in your nerve, you have a layer of fat that is called a myelin. And this myelin is important because it helps to speed up the information. And in Charcot-Marie-Tooth disorder, you can have forms that affect the neurons or forms that affect the Schwann cells here. However, I'm going to speak only about the forms that affect the axonal form here today. And particularly in what I call neurofilament disorder. Okay, so what are neurofilaments? Just to make a comparison that is relatively easy with your house, you have, if you have a house, you need a framework. If you don't have this framework, your house is going to collapse. That's exactly the same for a neuron. So you have a neuron here, and the framework of this neuron, the skeleton of this neuron, is going to be made by these neurofilaments. So basically, they are filaments, and they are here to strengthen the architecture of your neurons. So they are made of different protein, but that's a peculiarity of neurons. And what does it look like? So if you come to my laboratory and I bring you to the microscope, this is what you will see here. You will see a neuron with a dendrite, as I was describing you before. And you see here little filaments in green, which are representing neurofilaments. Okay, so they are making very nice neurofilaments. And in Charcot-Marie-Tooth disorder, you can have mutation in these neurofilaments, and this is one of the mutations, for example. And if you follow this arrow here, you can see that it's not the same as you can observe here. You have thick filament. It's like there's a bundle of filaments. So everything collapses in order to make a big bundle. And this is a problem because the structure of your cell is not going to hold properly, and your cell is going to collapse. So why I do that, call them neurofilament disorder? It's because there is a group of mutation in different genes that are linked to the same function. Okay, so you can have direct mutation in this neurofilament protein, and this is a Charcot-Marie-Tooth type 2E. Okay. And then you have a different functions that are related to neurofilament, like assembly, degradation, and that are involving different type of genes. And you find them in different form of Charcot-Marie-Tooth for peripheral neuropathy. So now what is interesting with intermediate filaments is that when you have a mutation in these genes involved here in how the filaments are going to be built, you can take fibroblasts from patients with carrying a mutation, and then you look at these filaments. And here you see, for example, this is a normal filament network. And here you see kind of ball in the middle of the cell. So all the filament comes back into the ball. And similarly, for this other type of peripheral neuropathy, you see another type of ball here. So all the architecture collapsed. And this is how we determine that finally a mutation is pathogenic, because the big challenge now is that with the next generation sequencing, we find plenty of mutation, but we need to find out if they are bad or not. So this is what I'm working on. And here I'm going to tell you a story about serendipity finally, and how sometimes it happens in the lab. We were working on this intermediate filament, doing basic science, and we were looking at what could bind these neurofilaments. And we found these two proteins, so the CK1 and the FAM83H. So the FAM83H is a middleman that bind to the neurofilaments and bring the CK1 close to the neurofilaments. And by doing this, then the neurofilament can change its behavior. And I was asking my student to perform an experiment on this on cells, and I say, well, do this on these cells. And when he come back to me, he said, well, I see something weird. And when you have a student coming back to you saying, I see something weird, is that just something that they see? So you go with them to the microscope. And this is the fibroblast I was telling you before. He mixed up the cells, basically, and he used the wrong type of cells. And he used cells from patients that are carrying mutation in the saxin gene or in the gigaxonin gene. And this is the famous middleman I was talking about, the FAM83H, that you need in order to recruit the CK1 alpha. And you can see the distribution here is wide in the cell. But then when you look at the cell of the patient, what you see here is that this middleman is collapsed in the center of the cell here. And it's obvious that there's something abnormal here. And that's where we starting to be concerned about this protein and their impact in the pathogenesis of this disease. So what does it mean that this protein is sequestered? We don't know, and that's what we want to find out, because it's important to us, especially if it means that the CK1 is not working anymore. And if it's not working anymore, then we can make it work using an agonist, okay, using a drug. So this is the hypothesis of the project that we proposed to the foundation, and we were glad to be funded. So we tried to find out what is the role of this CK1 and the middleman, this FAM83H, in the assembly of intermediate filament, and their role also in other type of disorder involving the collapse of this network. So I think it's quite important for us, because this is an example of how you can use basic science in order to move to translate to the drug development. Since we have this agonist that will activate this CK1, that will force this protein to work, we can try to use it as a drug to treat Charcot-Marie-Tooth type 2E, gigaxonin, and RSAX. So that's the novelty here. And instead of considering all this gene one by one, we consider them as a group, and we try to find out a drug that will work for all these different type of patients. So sometime you want to see big, and I mentioned probably that these neurofilaments are specific for neurons. They help for the architecture of neurons. But there's another type of intermediate filaments that are important. They are the same member of the family, but now they are specific for muscle. And they also cause a disorder in muscle when they are mutated. They are called desmin. And this desmin is quite important here in order to set up the stage for this Z disc here. And mutation in the gene cause a disorder, myopathy. So this is very interesting for us, because if we find a drug that could work on neurofilaments, on these filaments that are specific for neurons, would it work also for other type of intermediate filaments, like the ones that you have in muscle? And then this approach will be not only for patients that are carrying a mutation causing peripheral neuropathy, but also for this type of disorder. Thanks a lot for your attention. We never work alone, so there's a lot of people behind all of this. Sandra Minotti, Nancy La Rochelle, Alexandre Paré, Zachary, who are 2 students, and I have also other collaborators across the country and in different parts of the world. And I thank them for their support, including the support of the Muscular Dystrophy Canada. That was very important for us to start this project. And thank you for your attention. If you have any question, I'd be glad to answer them.
Thank you so much, Dr. Gentile. That was a really nice introduction to your work that you are working on. I just wanted to direct your attention to a question that we do see in the Q&A, and just being mindful of time, I invite you to respond by typing the answer. I also ask our other attendees to look at the answered questions. I know Dr. Kathari had answered some questions as well in the Q&A section. And then I will go ahead and introduce our next presenter. Next, we have Dr. Barnett Tapia. She is a clinician, scientist, a neurologist, and a researcher at UHN, University Health Network here in Toronto. And she will be speaking about her study, Patient Preferences and Treatments for Myasthenia Gravis.
Okay, can you hear me?
Yes, we can hear you and see your screen.
Thank you very much for MDC for this award. I'm really excited to tell you a little bit about why we're doing this research. And I'll just give you a view why we're really interested in patient preferences in myasthenia. So just some disclosures. So very quickly, myasthenia gravis, and we're talking about acquired myasthenia gravis. So this autoimmune disorder of the neuromuscular junction. And the classical presentation of myasthenia is fatigable weakness of different muscle groups, causing different degrees of disability and impairment in quality of life. And one of the good things about myasthenia is there are several treatments. And the cornerstone of treatment, this being an autoimmune disease, is either with some sort of immunosuppression or immunomodulation. So just to give you an idea, myasthenia gravis is called gravis because it can be quite severe. People used to die in a high proportion of numbers of myasthenia gravis when it was first identified. So in this graph, this is some history of medicine. In the 1900s, almost everyone, almost 100% of people with myasthenia died. And as we developed treatments such as tracheostomy and ventilation and steroids and thymectomy and ventilation and all sorts of immunosuppressants, mortality, this dark spots has decreased substantially. And now we consider that mortality due to myasthenia gravis is less than 10%. So it's a whole different world, it's a whole different disease now than what it was in the early 1900s. The majority of patients will improve with the treatments we have, although not everyone. About 10% of patients, so 1 in 10 patients will have what we call refractory disease. They don't respond to treatments. But even though most patients get better, very few patients come to this kind of holy grail of being in complete remission, having no symptoms, no signs, and being able to be off medications. So in other words, most patients control their symptoms, but will have some degree of symptoms with requirement of having medications. And this is just a way I organize medications or treatments for myasthenia gravis in my head, especially when I teach. And you can see we have lots of options now for patients, from treating just the symptoms, from actually modulating the immune system, for medications that we use kind of for rescue that are fast here, IVIG and plasma exchange, to more long-term medications such as steroids and non-steroidal options, classic treatments, and all this realm of biologic, which is kind of all the new trials in myasthenia are really in this realm of biologics. So we have lots of options, which is wonderful as a clinician and patients. If we had a drug that's 100% effective, meaning it treats all the signs and symptoms of the disease, basically you cure the disease, that works in everyone, not just in a subgroup of people, that has no side effects at all, and that's also cheap and easy to implement, and we would have a perfect drug, and we would not need to do any more research because we would just give this drug to everyone and we would cure everyone without a problem. But the truth is, this is not the case. None of our drugs are cures. They don't work in everyone. There are some groups of people. Some people do better with some medications than others. Everything has a potential side effect. And accessibility has been a key issue, especially with new drugs that are being available, are extremely expensive. So how to make that accessible, how to choose in whom to fund this drug, for instance, for our public healthcare system is a big issue. So because we have to address all these things, it's important to keep in mind that kind of trade-off and that balancing act of the medication we're giving versus the symptoms. So in my
do believe we lost connection with Dr. Barnett. Let me just quickly check in with her, and if not, we will wait for her to rejoin and go to our next presenter. Okay, I'm seeing that she is restarting. So what I will do is go ahead and pass this over to Dr. Voitel to introduce our next speaker.
All right, next in the roster, we have Dr. Mike Berger. In keeping with our theme of disorders, neuromuscular disorders, which are not genetic, but rather autoimmune, his project is looking at profiling granzymes in inflammatory neuromuscular disorders. He is a physiatrist and a researcher at the Vancouver Coastal Health Research Institute. Go ahead, Dr. Mike Berger.
Thanks. I'm just trying to share my screen here. Can you hear me?
Yes, we can hear you.
Good, good. Oh my God, there it is. Sorry. Hi, everybody. Thanks so much for having me today. I'm in the middle of a neuromuscular clinic today, so it's been very busy. I'm really glad that I could be here and tell you about this project. Now, full disclosure, I'm a clinician scientist. I'm a neuromuscular specialist, and my area of interest is actually in motor unit counting and electrophysiology. I'm not an inflammation scientist or molecular scientist. And I'll just tell you quickly how this project came about. I was carrying my load of boxes into my new office at the Spinal Cord Research Institute on my first day about a year and a half ago. And I was walking in like, I was a temp or something. And my next door neighbor happens to be Dr. David Granville, the office next door to mine. He's a world-renowned inflammation scientist. And David said to me, he said, Mike, what do you do? I said, well, I look after patients with neuromuscular disease. I said, what do you do? He said, I'm an inflammation scientist. And he goes, is there any inflammation in neuromuscular disease? And I said, well, you're damn straight there is. So we got to talking, and that was the germination of this project. And we wrote a grant, and kind of here we are. So David is really the expert on the inflammation side, and I'm the expert on outcome measurement in the clinic. So the project is about this unique serum protease called granzyme, and I don't know that much about it because I'm an electrophysiologist. So I'll do my best to introduce what a granzyme is to you today. But if you have any further questions, I might have to filter them through David. But I am able to tell you about this exciting project that we have kind of grouped together. And these collaborations are what makes for scientific discovery. You take people from different areas and mesh them together in a stew. And so we're really excited about this project. We have a couple of disclosures. David sits on the board of a couple of companies, and I was on the board of a pharmaceutical company. And here we go. So the immune system in neuromuscular disease, so, the body has a robust immune system that fights off infection, but sometimes it can get hyperactive and attack things you don't want it to attack. And in neuromuscular disease, the three things that you don't want it to attack are the nerves, the neuromuscular junction where the nerve talks to muscle, and the muscle itself. And so the three broad categories of inflammatory neuromuscular disease are inflammatory neuropathies, and most people here would have heard of Guillain-Barré syndrome, neuromuscular junction disorders, and you've already just heard half a talk about myasthenia gravis. Hopefully you get to hear the end of it. And then the inflammatory muscle diseases, dermatomyositis and polymyositis, are the most common ones. So the body's immune system gets ramped up and attacks these structures and makes them not work so well. And it can be very debilitating for people that have these conditions. So as a clinician who looks after patients with these conditions, it's a really frustrating group of patients to look after, and the patients are frustrated and the doctors are frustrated. And there's a number of reasons why they're frustrated. First of all, these conditions are actually difficult to diagnose. They're very rare in the grand scheme of things. There's often not a single blood test or a single imaging test or a single X-ray or a single anything that helps you arrive at a diagnosis. You often need the help of multiple specialists. So I'm a physiatrist. I often call on the help of my neurology colleagues or my rheumatology colleagues or my internal medicine colleagues to help me make the diagnosis. It requires a lot of investigations, and some of these investigations are invasive. If you think about a muscle biopsy, it's a minor surgery. So that's one challenge. The other bigger challenge, and the one that we're hopefully trying to address at least a little bit with our project, is that we don't really understand why the body's immune system flares and attacks these structures. So the nerves, the neuromuscular junction, and the muscles. So we don't know why the immune system flares, and we don't know what factors are responsible for autoimmunity. Now, within each of those conditions that I introduced, or the examples that I provided, we're getting better for sure. We're starting to hone in on what the actual targets are or what the factors responsible are, but we're not quite there yet. For me, my personal frustration is with the treatments, and the patients are also frustrated with the treatments. The treatments are problematic that we have for these conditions. So we treat inflammatory neuromuscular disease with immunosuppression. We try and suppress the immune system. So we can use steroids here. Maybe people have heard of IVIG, which is a blood product that we can use to suppress the immune system. There are old anti-rejection drugs. So people that had transplants and had to prevent their body from attacking the new organs. We borrowed some of those medications, and so those are called steroid-sparing agents. They're pills that we use to suppress the immune system. And there's new biologics that attack or suppress different parts of the immune system that we think are responsible for autoimmune neuromuscular diseases. There's a lot of problems with these treatments. They're really expensive, except for steroids. Those are cheap. They can be invasive, so you have to sit in an infusion clinic all day for IVIG or to get rituximab. There are a lot of side effects involved with these medications. You have to figure if we don't really have a good dial for suppressing the immune system. So we can suppress it too much and you can get infections. Steroids have horrendous side effects, including diabetes, bone disease, and cognitive side effects. The other problem with these treatments is, with the exception of the biologics, and even the biologics aren't so good at this, is they're not targeted. It's like putting a blanket on a fire, basically. So we try and suppress the immune system broadly, but we're not specifically targeting the causes of each of these diseases. So there's a great need to identify the factors that contribute to autoimmunity and to design safe and effective targeted treatments. We're starting to really make some gains in this field, but you know what? We're still the mountain climber at the bottom of the mountain looking up. So now I'm going to talk to you about granzymes. What are granzymes? Well, granzymes are a family of proteins that are secreted by autoimmune cells that have long been known to be a factor in immunity, but we're not really sure exactly how. There's about 5 of them that we study. Some of them work inside the cell, some of them work outside the cell, but there's some increasing evidence that these granzymes may play a role in inflammation diseases, although we haven't specifically looked for them very hard in neuromuscular diseases yet. Here's a schematic of what granzymes do. And you know, as a clinician, when I see these schematics, I kind of head to the washroom. It's like going to a concert and the band is playing all the new stuff and you want to hear the old stuff. But but we'll maybe walk through this a little bit. So these cells here up top are immune cells that can secrete this granzyme that then causes cell death. But granzymes also may work outside of the cell and do things like disrupt the barrier between the brain and the spinal cord and the outside of the body, and they can actually activate proteins that can cause nerve damage. And they certainly can increase inflammation in areas inside of the body. So there may be a number of roles of granzyme. And Dr. Granville has been instrumental kind of in the world in figuring out what the role of granzyme is. So Dr. Granville's actually done some research looking at multiple sclerosis, wound healing, blistering, some other immune conditions like rheumatologic conditions like scleroderma, and even in conditions where we think there's some autoimmune stuff going on, but we're not entirely sure. And so Dr. Granville has demonstrated that there's some increased granzyme activity in these conditions. There's not really a lot of I wouldn't say no direct evidence, that's maybe a mistake, but there's some very little indirect evidence that these granzymes are associated with human neuromuscular diseases. One study that's about 12 years old now looked at the thymus glands of patients with myasthenia gravis, and the thymus gland is, just generally speaking, has a role in the immune system, and certainly in some patients it's associated with the development of myasthenia gravis. And so they found evidence of increased granzyme being the thymus glands of myasthenia patients. I think it was 3 patients in this study. So this granzyme, it seems interesting. We should probably look to see if it's associated with some specific neuromuscular diseases that I look after. So our study purposes are to examine granzyme levels in patients with inflammatory neuromuscular diseases, but then determine the relationship between granzyme levels and disease. This should say disease severity. Sorry, I had some technical difficulties this morning. Here's our study protocol. We're looking at 3 of the most common neuromuscular diseases that affect the different parts of the neuromuscular system. And so we're going to look at Guillain-Barré syndrome. We're proposing 15 patients, just given where I work. So I work in Vancouver and Victoria in the neuromuscular diseases programs in both cities. And just based on the number of patients that we see on an annual basis, that's how we selected the number of patients that we expect to be able to examine. So Guillain-Barré 15, myasthenia gravis 15, and inflammatory muscle diseases 15. We're going to look at certain tissues where there may be, where we can often measure inflammation or antibodies. So in Guillain-Barré, we're going to look in the cerebrospinal fluid and in blood. In myasthenia, we're going to look in blood. Maybe we may or may not find anything in there because blood may not be the best place to look. But in myasthenia, we don't really diagnose the condition on anything other than blood work. So it would be a bit invasive for patients to take chunks of muscle or to look at their cerebrospinal fluid. And then for the inflammatory myopathy, this may be the biggest bang for our buck here, is because we actually routinely get muscle tissue to make the diagnosis clinically, although not as much anymore because we have really good blood tests for inflammation muscle diseases, but we still on occasion will take a muscle biopsy. And we're also going to look in blood as well. We're going to compare the tissues in each of these disease conditions we have a biobank of healthy control tissues. So we have healthy people's cerebrospinal fluid, muscle, and blood. And so we're going to compare granzyme levels to each of those conditions. But then from my perspective, the thing that's most exciting for me is I spend a lot of time looking at outcome measurement in neuromuscular diseases and other neurological conditions. So what we're going to try and do is we're going to try and determine whether the level of granzyme activity is associated with how severe the clinical presentation is. And we're going to use some these are all the EGOS, QMG, and IMAX core set. We could spend days going into what each of those are, but basically they're just battery tests that give us an overall score of disease severity. So that's what we're proposing to use as our measure of disease severity. I'll tell you where we are right now, obviously, and I think I've been in and out of the talks all morning because I've been seeing patients, but my understanding is that everybody's had some challenges with COVID. We were no different, so we did have to delay our funding a little bit. We got our funding from MDC in September 2020. We now have ethical approval, and we have completed our COVID research resumption plan because each of our institutes and each of our clinics needs a research resumption plan approved by the university. So we're just waiting until next week for our plan. We suspect it'll be approved because we're not doing anything that's outside of routine clinical care. So we expect that our research plan will be approved, and then we can begin data collection in December or possibly January 2020 that will take us through to the end of August 2022 when our funding expires. So, you know, we have no preliminary data to share with you, unfortunately, because the whole idea is very preliminary. This is very exploratory, but potentially very exciting. How will this help the people in the audience today? And how will this help people like me? Well, at minimum, it's going to improve our understanding of autoimmunity and neuromuscular diseases, which is so important towards just our understanding of these conditions in general. We may, because granzyme is kind of a hot button protein, find a biomarker of disease progression, so that if granzyme levels are elevated and the higher levels are associated with worse disease severity, we may be able to monitor granzyme as a marker of response to treatment or disease progression. And because granzyme is associated with so many potentially bad things in autoimmunity, it's a potential drug target. So with that in mind, kind of that's where we are, that's our project. This is who we are. You can't do a project like this without a lot of help, especially on the clinical side. So Dr. Granville and his lab are very well established, but on the clinical end, we have lots of help. Dr. Peter Schutz is the neuropathologist who's going to help analyze the muscle biopsies. And then these are all of our clinical partners in Vancouver, lovely Vancouver and lovely Victoria, where I am right now. And so these are all the people that are going to help us with clinical outcome measurement and recruit patients. So we're very excited about this project, and we're very thankful to Muscular Dystrophy Canada for providing us with funding to get this project going.
Thank you very much. And thank you very much for a clear presentation, for the work that you and Dr. Granville are doing with this project. I also want to thank you and the other panelists for modifying their clinical schedules, research schedules to attend this session today. Thank you so much. How about, given that the topic is so close to myasthenia gravis, Dr. Barnett Tapia has rejoined. We'll pass it back to you, Dr. Barnett, to continue your presentation. And we'll ask that the video be off, and that might help maintain a stable connection.
Yeah, apologies. I think I'm not allowed to share yet. I think the other screen has to be.
Yep, Dr. Musburger, you'll be able to stop sharing on your end, and then we'll have.
Yeah, okay. My apologies. I got kicked off. Okay, so going back to where I was. We're talking about trade-offs, and I'm glad we had the previous talk about how untargeted our treatments are, so they tie really nicely. So these are some of the common symptoms that patients with myasthenia complain about: double vision, droopy eyelids, difficulty swallowing, difficulty chewing, difficulty pronouncing words, poor level of voice, reduced facial expression, shortness of breath. In bad cases, can go to intubation, weak arm, legs, and neck. So as you can see, they can cause a whole set of restrictions in the activities of daily life and the ability to care for your family, for working. It can cause all the different levels of disability. But even though we have lots of treatments, they have lots of potential side effects, and some of them are non-trivial. So whenever you suppress the immune system, you have increased risk of infections, which can be severe and lethal. There's an increased risk of cancer. It's a small increase, but it's a true increase of risk of cancer in long-term immunosuppressant medications. People can get diabetes or worsen diabetes, high blood pressure, and therefore you can have increased risk of myocardial infarction and cardiovascular disease. You can develop cataracts, osteoporosis, fractures, and all sorts of toxicity that we need to monitor, weight gain, etc. So while it's great to have treatments, and our patients have lots of symptoms that need to be improved, we need to help them so they can do their daily life. Our treatments come with all these potential adverse events, and it's not always easy how to make a decision because of this. So how would someone make a treatment decision? So basically, it's like any decision in life. You kind of weigh your pros and cons. So this is a theoretical example. You could say medication A might improve my double vision, give me more strength, reduce my likelihood to be admitted to hospital, improve fatigue. Maybe it's very convenient. I just need to take one pill a day and I'll be able to move more. But on the con side, I will probably gain weight. I have the risk of severe infections. I might have this increased cancer risk. I need to get my blood checked every month. Maybe it causes hair loss, and to make matters worse, it's expensive. How do you make that decision? And it's a very individual choice, but there's ways to systematically assess how people in general make choices. And in clinical practice, sometimes we see patients who we think, oh, this drug would be perfect for this patient, but the patient is adamant they don't want it. And sometimes what seems irrational to a clinician is really that a patient, a given potential con, say, I'm just going to say maybe weight gain, is a deal breaker. That attribute is so important, it weighs so much that it takes away any potential benefit. And clinicians, we do this too. We think, oh, for this patient, pros and cons, and we also have bias because we're human beings. So this whole way of making decisions is a human nature thing. And we have no idea how people with myasthenia make decisions, and we don't know systematically how clinicians make decisions. We know from other diseases that the way clinicians and patients make decisions can be very different, and they don't always see face-to-face. And I think that's a problem when we're not doing shared decision-making and we're offering things that may not be what patients in general are more interested about. And as I said, we have no data from fatigue, but we know from other studies that we've done previously in myasthenia that patients view things differently. And I'll give you the example of fatigue. So we've developed a questionnaire to assess symptoms of myasthenia gravis called the Myasthenia Gravis Impairment Index. And when we interviewed patients for this, fatigue and this concept of having weak muscles and getting tired as the day went by was extremely important. But then when we sent the questions to experts to comment on the items, patients, the experts were really like, oh, why you can't ask about fatigue. I mean, everyone is fatigued. The patients don't understand it. The patient is very subjective. The patients cannot make a difference between fatigue that's due to myasthenia or not. And when we came back to patients were saying, oh, no, this is extremely important for us. So we found this major disconnect. And we decided to keep the patient's perspective in our measure. And as time has gone by, we've shown that it's actually very important. Some of these biases from the experts were not true. Patients can indeed differentiate. Not everyone is fatigued, and fatigue actually can improve with treatment of myasthenia. So we know, we've shown that the views are very different, and we want to learn more a little bit about that. This is another thing we've done when we've seen a way of patients evaluate their health. So this is a way where people who feel their health is perfect score up here, which is 1, and if you have pretty bad health, kind of almost being dead is 0, and you can have negative values. When people are really ill, they feel their health is worse than being dead. And we looked at symptoms of myasthenia, and of course, as symptoms were worse, patients were worse. In this study, having weakness in the arms and the core and the neck was what was driving the health quality, the quality of life for patients. However, when you ask experts to weigh on symptoms that are more severe, they will always say that this bulbar, so difficulty swallowing and breathing, are more important. And I think it comes from different purposes. As clinicians, we're always worried about patients who may need to come to the ICU and be intubated. And that, I'm not saying it's not relevant, but when you look at people who are in the community, outpatients, the ability to move around, the ability to do their regular things becomes dominant. And sometimes we forget about that. If the breathing is fine, we may not treat these other things as well. So this is why I think having the patient's perspective is so important. So this is where a project comes from, and it's called a DCE, that stands for Discrete Choice Experiment. And this whole methodology actually comes from like behavioral economics, is the way people make choice. And this is the gold standard to understand. So in this kind of framework, an economist will say, you will only know what someone really, really values something when you make them choose. So if you ask someone to make a list of things that they care about, but then that doesn't always translate to what people choose. When you force someone to choose, you know what really drives their decisions. So we're making this where we'll do a survey to understand how people and living with myasthenia make these trade-offs between potential benefits and potential side effects from treatments. And we will also analyze clinicians. So now we're delayed with COVID as everyone else, but hopefully we'll have our REV, and we'll start interviewing patients virtually soon. So it's all going to be doing virtual qualitative interviews. And what we try to get first is kind of idea of what are the major pros and cons that most people are interested in. And with that, we will develop a survey. And this is just something I made up. So this is not the real survey, but this is what it would look like. So we will come with some treatment characteristics that I just made up for this example, as say mode of administration, infusion reactions, severe infections, and some symptoms. It might look like something else. And there will be several levels. So patients will be presented with different scenarios. Say, if you have to choose between treatment A and treatment B, choose which one would you choose if you had no other options. And we will vary each of these. So there's hundreds of possible combinations, and I'm not gonna get you bored with the math, but there's a way to make this really efficient and optimize the information with the minimal amount of scenarios. But people are forced to choose. And after analyzing lots of people, you can see what kind of what we call attributes are more important. So it may be that severe infections, the risk of severe infection is a driving attribute, that people really want to minimize severe infection. Maybe I'm wrong, maybe it's going to be weight gain, or it may be time that you require to take the medication, but that's something we will find out. So this is something that hopefully we'll be able to do in the next year. And if you're thinking, why is this important? This is important because we need clinicians to think more about the patient's view. So in clinical practice, we're supposed to do one-on-one, but if you look at clinical guidelines, there's zero input from patients. In all the clinical guidelines for myasthenia, they all come from experts. There's nothing with the patient's view. This is also important for decision makers. Say when we discuss in our public healthcare system which medication we should fund, yes, efficacy is important. But if we have evidence that certain profiles of medications are better from a patient's perspective in terms of the convenience and the side effect profile, that's also something that decision makers take into account, even if it's an expensive drug, for instance. And also it helps development. So if we find that a certain side effect is really bothersome for patients, it might help pharmaceutical companies to come with ideas how to improve on that profile and develop drugs that minimize those potential side effects. So this results, we hope, will have clinical impact at different levels. So with that, I'll leave you. I'm happy I'm still connected. Lots of people to thank. This, part of the work we've done, and this is a really collaborative effort. Ahmed Bayumi, who's not a neurologist, but he's an expert in decision making. So he's really helping us with all the methodological issues. And of course, Muscular Dystrophy Canada, because we couldn't do this without the funding. So we're really, really, really thankful. MGNet is collaborating with us, which is great. And Myasthenia Gravis Foundation of America, this is a patient, also a patient organization focused on myasthenia, and they're the ones who are going to help us expand our survey. They have thousands of patients registered, so hopefully we'll be able to get a very large number of respondents. And for the sake of time, I'll leave it there.
Thank you very much. I welcome attendees to type in their questions in the chat box, and Dr. Barnett Tapia can comment there. This is important work and in line, and it reminded me of a project that we have right now here in Ontario with MDC and London Health Sciences around shared decision making. And so what you've highlighted is how important that patient voice is when making a choice, and we look forward to the results of the study. I'll pass it over to my colleague, Dr. Voitel, who will introduce our next speaker.
Next up, I'm happy to introduce Dr. Vladimir Lubavitch. He is an associate professor at McMaster University, and his talk is titled Reverse Translation from Diabetes Clinic to Preclinical Muscular Dystrophy.
Thank you. Can you see my screen? Wonderful. Thanks for the introduction. Thanks everyone for being here this afternoon in this session. And a special thanks to Muscular Dystrophy Canada for providing this platform to share with the community some of the work that we've been doing, investigating the neuromuscular system in health and disease. So in this 8-minute thesis, what I'd like to do is provide a brief introduction to the people in our lab who do the work, and as well as some of the research that we have ongoing. And then also provide a short summary of the research grant that we were really fortunate to be successful in acquiring in the last round of the research grant competition from Muscular Dystrophy Canada. So this is the team in the laboratory. We have a wonderful group of trainees, PhD students, master's students, and undergraduate students. And many of these folks are going to go on to become physicians. They're going to be independent scientists. They're going to be allied health professionals and educators, and they all have a passion in understanding the neuromuscular system. I want to give a special shout out to our laboratory manager, Todd Pryor, who keeps things running safely and smoothly in the laboratory, particularly during the COVID climate that we're all experiencing right now. We're really fortunate to have wonderful collaborators, local collaborators at the university that assist us with many of our ongoing projects. And so it's a pleasure to acknowledge all the contributions from the team. And also, I'm always happy to take a moment to say thank you to our funding partners who help make our work possible, in particular, Muscular Dystrophy Canada. Our laboratory is located in this building here. It's in the east wing of the Ivor Wynne Center at McMaster University in Hamilton. It's located right beside the athletic complex, and it's a short walk away from McMaster Health Sciences and the Children's Hospital, located just on the other side of campus. We're part of the Faculty of Science, and our laboratory is located in the Department of Kinesiology. And kinesiology is the study of movement, health, and disease. And our lab is part of a cluster of laboratories in the Department of Kinesiology that have a common interest in understanding exercise and metabolism, as well as heart function and muscle function. And together we're called the Exercise Metabolism Research Group. Our laboratory specifically is interested in understanding the factors that determine and maintain and remodel the neuromuscular system in health and disease. And we're really interested in understanding the role of a particular enzyme in the neuromuscular system. And this enzyme is called AMP, activated protein kinase, or AMPK for short. And AMPK is a protein. It's expressed in numerous tissues in our body, and it's critical for a number of different functions. And for example, in the brain, AMPK is important for regulating appetite. It's important for the heart to maintain the fuel for the contracting muscle cells. It's also important in the liver to maintain proper liver function. Now, we and many others in Canada, and in fact, many other laboratories all over the world, are interested in studying AMPK in skeletal muscle. And we're really interested in understanding whether or not, or further examining the therapeutic potential of turning on this protein in different neuromuscular disorders, and for us specifically, muscular dystrophies. And that's where our work with Muscular Dystrophy Canada comes in. We have a few studies on the go that they are very kindly supporting. And in those studies, we are using a new strategy to turn on this enzyme and to understand whether or not it's an effective therapeutic target for muscular dystrophy. We're using a new compound, a new drug that is practical, so it can be taken orally as a liquid solution. There are no injections or infusions necessary. It's safe in type 2 diabetes patients who have been treated with this compound. There is no negative secondary effects that have been identified so far. And it's effective at improving clinical outcomes in patients with type 2 diabetes. And it's also effective at turning on AMPK in the skeletal muscle, which is critical because this is the tissue where the enzyme needs to be activated if it's going to have any impact for muscular dystrophy. And so we have two studies ongoing. In the first study, we're looking at the effects of a single dose of this compound in mice with muscular dystrophy. So we have a number of animal groups in this study, and we're using a preclinical model of muscular dystrophy, both of myotonic dystrophy type 1 and Duchenne muscular dystrophy. And these are established animal models of the human disorder that mimic quite closely, with some limitations, what's happening in myotonic dystrophy type 1 patients and in DMD patients. So in this study, we have a number of different groups. We have healthy animals that receive a single dose of the placebo solution. And we have healthy animals that receive a single dose of the experimental solution that has the compound that turns on our protein of interest. And this compound is called O304. And we acquire this compound through a strategic partnership with some of our industry colleagues. And we treat these animals, and 3 hours after we treat them, we take their tissues and we perform analyses. We have other groups of animals that have muscular dystrophy. And in some of those animals, we give them the placebo solution. And in other groups, we give them the experimental solution, and we take their tissues 3, 12, or 24 hours after a single dose. And we have some very preliminary data to share that I'd like to share with you today. We observed that 3 hours after a single oral dose of this compound in dystrophic mice, our protein of interest, AMPK, was in fact turned on in the limb skeletal muscles, so muscles of the lower legs, as well as in the diaphragm of these animals. And it's really important that the enzyme was turned on in the diaphragm because we know that this tissue is severely affected in patients with muscular dystrophy. Not only was our protein of interest turned on, but there was also an increase in a number of cellular events, including the expression of some genes, so the expression of the DNA that we know has been proven to be beneficial to reduce the severity of muscular dystrophy. Some genes were turned on, but not all the genes that we anticipated were turned on at this point. But we're not too disappointed because although the experiment is completed and all the animals have been treated, we're still analyzing many of the samples from some of the time points. So this study will tell us how this new compound is working in muscular dystrophy, in animals with muscular dystrophy, but it won't tell us whether or not turning on our protein of interest using this new compound is effective at reducing the severity of muscular dystrophy, at preserving muscle function. And so we have a second study that's ongoing, where instead of treating the animals only once, we treat them every day for 6 weeks. And we assess their muscle function. We look at the muscle endurance. We look at their muscle strength, their ability to move around, their enclosures. We assess heart function, and we perform a number of measurements on the muscle cells, at the neuromuscular junction, and the nerves in order to determine the effectiveness of this compound at preserving muscle health and function. So together, these studies will give us an indication whether or not this new way of turning on our protein of interest, AMPK, using this new compound, is safe and effective at reducing the severity of the disease. And it'll give us an indication of whether or not this compound has the potential to translate into patients with muscular dystrophy. And so I want to thank Muscular Dystrophy Canada one more time for giving me this platform. Thank you all for your attendance and your attention. It was a pleasure for me to introduce the lab team and a little bit about our research. And also, we're really grateful for the support from Muscular Dystrophy Canada, the support for the students in the laboratory to do this work that we think is going to have impact for patients with muscular dystrophy. Here's my email address. Please feel free to send me any comments or questions that you may have at any time. And I look forward to carrying on this conversation. Thanks a lot.
Thank you very much. Thanks for a great presentation and for sharing those preliminary findings with us. We'll turn it next to Dr. Alex Flynn, who's a postdoctoral researcher working with Dr. Michael Rudnicki at Ottawa Hospital Research Institute. Today, Dr. Alex Lynn will be talking about a partnered grant with CIHR, a multidimensional single-cell approach to understand muscular dystrophy. Dr. Lynn, we don't hear you quite yet. I can check to see if I can support with the audio.
Sorry. Okay, there we go. So sorry, just a big picture overview. So the adult human, you have a lot of organs and tissues that are able to regenerate. And just shown here are just some of them. And this ability to repair your injuries comes from a specialized population of cells known as adult stem cells. And in Dr. Rudnicki's lab, we're really interested in skeletal muscle. And you can see here, the satellite cell, also known as a muscle stem cell, upon an injury is basically able to repair all this muscle. So you can see here in uninjured muscle, this is how it kind of looks like. When you damage it, you can see all those myofibers are all gone. And then by 21 days after this kind of damage, it is completely repaired. And this is all due to these satellite cells or muscle stem cells. And so in our lab, we're really interested in characterizing the genetics of this and how this is going aberrantly in kind of disease conditions. So how we understand how muscle stem cells make your muscle tissue is this very linear process. However, your muscle tissue is very diverse. So your arm muscle is very different from your leg muscle, but how we still understand it is this very linear process. So how can you account for this diversity seen at the muscle tissue if it's very linear? And we believe that at the muscle stem cell level, that there is already heterogeneity. So there's distinct cells that may be undergoing different paths towards making muscle tissue. And indeed, previous work in Dr. Reniki's lab has shown that a specific population of these muscle stem cells undergo asymmetric and symmetric division, so that is able to generate, maintain self-renewal and also generate progenitors. But also more importantly, this ability to undergo asymmetric cell division is mediated by dystrophin. So in the absence of dystrophin, what we've also shown is that there's stem cell hypoplasia and they don't generate the proper number of progenitors to repair the muscle tissue. So going forward, our question was, are there other distinct functional subpopulations within this bulk population of satellite cells? So perhaps there's something that sits on top of all this hierarchy and can actually be the muscle stem cell that we should be focusing on. So those are the two main questions. Can we uncover more of these populations, and can we target them? So to do so, we took this kind of multi-dimensional approach, I guess you could call it, but it's mainly using single cell genomics. So you can see here this bulk population of muscle stem cells. If you have a rare cell type, it will be diluted out when you assess them all in bulk. So you don't get to understand rare subtype cell populations, and the heterogeneity is masked. But with new advances of single cell technologies, you can take individual cells now and assess the distinct gene expression of each individual cell. And perhaps we can then uncover disease-driving subpopulations or ones that are more stem. So an analogy to this is previously we're looking at a fruit smoothie, but now we can look at this as like a fruit salad. So picking out each individual piece of fruit and trying to assess what it does. So to do so, we started off using healthy wild type mice, and we sorted these mice, captured this on this 10x genomics platform, which is the single cell platform, did the sequencing and bioinformatic analysis. And when you do that, you get these kind of really nice plots that come out. It doesn't really matter too much what all of them mean, but each individual dot is are these individual muscle stem cells. So again, previously technologically, we weren't able to uncover all these different color groups, and it would all just be masked. But now what we can do is kind of trace these cells out, and what we believe is happening is that we've identified this novel subpopulation sitting at the top that has never been characterized before. So if we were to take more bioinformatic approach to understand what are the molecules or genes that are important in this novel subpopulation, we can identify a lot of them. So you can see these are the different genes or molecules that are upregulated in this novel subcluster. The intensity of green means that they're really enriched in this population. And we focus on integrin alpha 1, or also known as CD49A, shown here. So does this subpopulation actually exhibit any unique characteristics? So to do so, we did the gold standard assay of stem cells and did transplantations. So to do so, we isolated these cells in an initial transplant, transplanted them into mice, and then assessed their re-engraftment. So all these cells that were isolated were GFP labeled, so green fluorescent protein. And then one characteristic of muscle stem cells, or stem cells in general, is that upon multiple injuries, these cells will persist over time. And that is a characteristic of stemness. So in our initial transplant, we would kind of predict that there would be more GFP fibers, or these green fibers, and also PAX7 positive cells, which are those muscle stem cells. And upon re-injury, we would predict the same thing, indicating that there are truly a self-renewing population. So in the initial transplant, what we can see is in the negative population, that CD49 negative, so not the novel subcluster, there are green myofibers that are formed. There's not many muscle stem cells, but upon transplantation of this CD498 positive muscle stem cell, you can see way more GFP or green myofibers and a lot of muscle stem cells that have been re-engrafted. When you re-injure, again, there's not many in the negative population, but there's a lot of persistence in this integrin alpha-1 subpopulation. And that's quantified over here. So in this initial transplant, you can see there's a significant increased number of fibers and also satellite cells or muscle stem cells. And then upon re-injury, we see the same thing, indicating that this is truly a self-renewing muscle stem cell population and it has enhanced engraftment. And it's also really interesting is that this subpopulation, although we started off with wild-type healthy controls, they comprise of a very minute population of the total muscle stem cell. But in a MDX mice, which is a model for Duchenne's muscular dystrophy, we see that it's significantly upregulated or it comprises a much larger subpopulation. And this is really interesting going forward because we've previously shown that in normal conditions, there's a balance of asymmetric and symmetric divisions where you get progenitor expansion and self-renewal. But in DMD or in the MDX mouse, you have this stem cell hyperplasia. So if we have now identified a molecule that is kind of this causing this stem cell hyperplasia, maybe we can then target it. So just with all this preliminary results that we have so far is that we use this single cell sequencing approach and we've identified a novel subpopulation of muscle stem cells and that this subpopulation shows enhanced engraftment and stemness. Going forward, we're really interested in doing more multidimensional approaches such as proteomics and more sequencing to understand what molecules give this subpopulation its interesting characteristics. Maybe we can then identify drug targets that can then mediate the division kinetics to ameliorate dystrophic conditions. And then ultimately, using this single cell approach, I think I've shown that this pipeline is really powerful to identify a lot of different cell populations. And so we want to integrate this with a lot of different genetic backgrounds. And so, for example, the MDX mouse model, how does that look like on this, with all those single cells? So with that, I'll thank the Renicky Lab, a lot of help, and then all the funding agencies. And I'll be happy to take any questions.
Thanks so much, Dr. Lim. That was an excellent presentation. A common question that comes to our research hotline is what is the updates in terms of stem cell research? So thank you for providing a presentation on this topic. I'll turn it over next to Dr. Erin Beattie. She is the network manager at the Neuromuscular Disease Network for Canada. I would also like to thank all of you for staying past the hour. This is our last presentation. We're really grateful to have Dr. Beattie here today to share the updates on the Neuromuscular Disease Network for Canada. Over to you.
Thanks, Mira. And you can see the full screen. Everything's okay?
Looks great. Go ahead.
Okay, perfect. I'm going to start my video, but if the quality is poor, I'll just go in and turn that off.
Perfect.
So thanks for inviting us to this opportunity to tell you everybody a little bit about our research network, the Neuromuscular Disease Network for Canada, NMD4C for short. So a little bit about what we do and who we are. We're a new pan-Canadian network that brings together the country's leading clinical, scientific, technical, and patient expertise to improve care, research, and collaboration in neuromuscular disease. And we launched earlier this year with funding for a 3-year funding from CIHR and Muscular Dystrophy Canada. And we really work on networking, collaboration, community building, capacity building, knowledge translation, and data and sample access. We really are involved in supporting and developing resources, such as supporting patient registries, such as the Canadian Neuromuscular Disease Registry, the CNDR, developing resources such as a virtual biobank of really a catalog of samples obtained from patients with neuromuscular disease, and initiatives around KT initiatives, so developing patient-friendly clinical guidelines, developing resources on our website, hosting annual conferences, including patient days, which we're hoping to do in person next year, and also training and education initiatives, such as our genomics rounds, and we've developed a monthly webinar series, and really we hope after our 3 years that our impact would be to really attract more clinical trials to Canada, attract more public and private investment in neuromuscular disease, and really support the next generation of clinicians and researchers, and really to create sustainable resources for future research and sustainable community of patients and professionals, and have improved diagnosis and access to therapy and improved information for patients and advocacy organizations. So we have a great team that governs the network. This is our steering committee, and Dr. Hans Lockmuller leads the committee, and along with Drs. James Dowling, Jody Warman, Chardot, and Rashmi Kathari and Stacey Minturn. We also have a KT lead, Dr. Cynthia Gagnon, and really they work together to provide overall scientific leadership and direction and oversight of the network. They really represent our patient advocates, our researchers, and our clinicians. And we've recruited almost 300 members, spanning really across Canada, from Victoria all the way to the East Coast in Halifax here. And we've had continued growth from really conception in January to now, where we're about at 293. And we have the majority of our members are researchers and clinicians, patients and family members of those with neuromuscular disease, and a minority from biotech, pharma, patient advocates, and healthcare professionals. We've developed quite a few resources on our website, and this is just a snapshot of our homepage. We have a little bit more about who we are and our team members. So our steering committee, our investigators, our collaborators that really do all the work on our various initiatives, and more detail about our entire work package, what we hope to accomplish over the 3-year period, an area where we hope to recruit more members. Also news items, where we have, this is showing you a little snapshot of what we have offered on that part of the website. So where we have our monthly newsletters, and promoting upcoming events such as our webinars and any other kind of items of interest to the neuromuscular community and our membership. We also have an area of our website dedicated to our webinars and our early career blogs. So I'm going to go into a little bit more detail on these last two. So in collaboration with MDC, we have one of our main goals is to really train, educate the next generation of neuromuscular disease clinical specialists. And we do that one way of, we've started an accredited monthly webinar series in collaboration with MDC. And this is the first Tuesday of every month from 5 to 6:30. And really so far we've had topics such as the practical considerations for clinicians. So updates really of the state of neuromuscular disease. So one on SMA and one on Duchenne. And this was done in partnership as well with Jesse's Journey. We also have a focus on genomics training. So we had sort of an introduction to genomics, so Genomics 101, and a follow-up with diagnostic techniques for genetic neuromuscular diseases. And we really have want to continue this focus on our genomics training, and we will have further genomics webinars quarterly going forwards. We've also had basic science sessions, such as the genetics of congenital myopathies in French Canadians. And after our webinars, we also developed these webinar reports where we have sort of the key take-homes from the webinars, and those are posted onto our website. We also want to empower really and support our early career researchers and clinicians. And we do that by involving them in our webinar sessions. So our next webinar is on Tuesday, December 1st. where we have really our up-and-coming stars in neuromuscular disease. We have 4 of our early career members that are going to be presenting, and actually one is going to be chairing the session as well. So if you'd like more information on that webinar, it's online on our website, which is listed at the bottom of my screen as well. We also have around once monthly, we have blog posts from our early career members. And our most recent one is from our early career researcher, Dr. Melissa Bowerman. We also are going to be hosting our first early career forum, and this will be virtual at the end of February. And so more details will be available on our website in a few weeks. And we're hoping to offer these 1-week clinical placement opportunities, but that initiative is really on hold until sort of post-pandemic. And how you can get involved, and if you'd like to stay up to date on what we're up to, one avenue is to subscribe to our newsletter. So on the left is showing an example of our most recent newsletter in November. You can also follow us on our social media channels, such as Twitter, Facebook, LinkedIn. We also have a discussion forum on Slack for all of our members. It's really facilitating communication between our members and between our investigators that are working on all our initiatives. You can contact me if you have any questions at any time, or also through any of these avenues here and our website as well. And if you'd like to register and become a member and really tell us how you'd like to be involved and participate, we would love that to hear from you. So I want to sort of thank everyone, including Muscular Dystrophy Canada and CIHR for the generous funding, and really to all of our investigators and collaborators who are working on all our initiatives. This is just a snapshot from our first annual meeting that just occurred this week. So thank you very much.
Thank you very much, Dr. BT. That was a great introduction. What I'll also do is in a follow-up email to all of our participants today, I will share the network's website and how you can join the network as well. We have come to the end of our presentations. I'll pass it over to Dr. Voitel, who will share some closing remarks on. I would like to thank everyone for staying and for appreciating the fact that we're doing a little bit of a tag team here and moderating today's session. So over to you, Dr. Voitel.
Thank you, Homera. Thank you to all of our speakers and their research teams. I think that's one of the key messages we heard today. It takes many people to move this research across the line, and these sessions have really highlighted the exciting work that is to come. And it wouldn't be possible for us to be able to fund this research without our generous donors and supporters. So thank you to that group as well. I also like to thank all of the individuals with lived experience who motivate and give direction to this work. And we are looking forward to launching our next grant competition and showcasing the next awardees in 2021. Stay tuned for a next session like this in the new year. Thank you so much.
Thank you very much. To end the session, I welcome you all to join our day 6 of the Virtual Neuromuscular Conference. It will be on Tuesday, November 24th. Dr. Voitel had mentioned one of our past awardees, Dr. Leanne Ward, will be talking about bone health and neuromuscular disorders. As always, I'd like to thank PTC Therapeutics, Roche, Biogen, and Alexion for supporting education initiatives like this. Again, thank you so much to our panelists for taking the time to share their work. We know COVID has caused a lot of disruptions to them starting their projects, but the fact that they were all enthusiastic to join us today to share the project has been incredibly valued. Thank you all so much and have a great weekend. Take care.